So thanks, Krishna, for a nice introduction, and very fortunate to be here today and present our recent work on a particular pathogen called Enterobacter bugandensis and its adaptation in International Space Station. So I'd like to start with acknowledgement. I'd like to thank my supervisor, Professor Karthik Raman at IIT Madras, and Dr. Kasturi Venkateshwaran from JPL, and my co-authors Shobhan and Nitin. And we just recently published this paper in Microbiome. So let's just jump into today's topic. So very fast, why we are interested in space microbiome? You probably have imagined this from the title, because there is ongoing lot of efforts on human habitation in Mars and beyond. There is SpaceX and other space agencies like NASA and other agencies are putting a lot of effort in understanding what is the life beyond the Earth. And to understand this, we the very good place to understand this is an International Space Station, which is now in the space over 25 years. And why this is important? Because it is a built environment and it experiences a lot of extreme phenomena, like there is microgravity, there is a lot of CO2 level, and a lot of space radiation. And definitely this place is not micro free. This has a lot of microbes, and because there are astronauts and there are ongoing cargo, and this causes a lot of microbes to be there. And under those trays, how these microbes are adapting there is something that we are interested in. And then there are also pathogens there, and then the question is how we develop a countermeasure against them. So we need to understand this pathogen and then potentially drug development to deal with these pathogens. So NASA does a lot of microbial tracking mission to track these organisms time to time, and this one of the microbial tracking mission has produced nearly 100 bacterial strains that are isolated from International Space Station. And upon sequencing, we got to see that there are 12 Enterobacter bugandensis. Why we are particularly interested in Enterobacter bugandensis is a BSL level 2 pathogen, and it's a nosocomial pathogen which is usually seen in hospitals and particularly affect neonates with sepsis. So that's why we are interested in Enterobacter bugandensis. And we tried to ask two very simple questions. Number one, how they are different, how they are adapting to the International Space Station extreme environment, and what genomic differences they have from their Earth counterparts. And secondly, how they are shaping the microbial diversity in the International Space Station. So to address these steps, we took two-fold approach. Our first approach is a genomic characterization. We got all the genomes we had during that time in the NCBI. We did genomic characterization, pan-genome analysis. And in the second axis, we analyzed metagenomic samples obtained from International Space Station during 2016 to 18. We identify what are the co-occurring community with the Enterobacter bugandensis, and what kind of metabolic exchange is happening, and that is shaping the community over the International Space Station. So our very first observation, when we build a tree, we notice that the strains from International Space Station is forming a distinct and separate clade from other species in the on the Earth. And then that is very, so they are genetically different, slightly different from what are the species we usually see here. And this is again another method using SNP and mutations. We created this tree, and this also corroborate with what we seen in the last slide. And you can see the strains from International Space Station have a difference of nearly 36,000 SNP from its type strain EB247. And even with its closest strain AR1358, this is nearly 3,000 SNPs and nearly 4,000 mutations different. So that indicate that these strains that we got from International Space Station are genetically different from the strains we see in the Earth. Then we try to say, okay, they are genetically different. What about functions? So we identify the functions. These are functional gene clusters, which is shown here as a heat map, where red indicate the presence and blue indicate the absence. And each row is basically a genome. And on the clustering, you can see International Space Station strains. Again, becoming a clustered, unique cluster here. That says they are not only genetically different, they are also functionally different. And then what is making them different? So there is a patch of genes which is very unique to International Space Station, and we try to see what are they. So they are related to stress associated genes, they are associated with transcriptional regulator, SOS response, which all says that they are under stress in the International Space Station, which is usually not seen in the other earthbound strains. And in general, the International Space Station genomes having a more number of genes, on an average more than 100 genes when we compare to the Earth strains. So this says that International Space Station strains of Enterobacter bugandensis are very different from its earthbound strains. But these are isolates. What about their abundance in these organisms, abundant in the International Space Station? So for that, we collected 42 metagenomic samples across 3 time points of International Space Station within the time frame of 2016 to 18. These 42 samples were distributed in 2 groups. One is treated with PMA. PMA is propionodium monoazide, which identify the viable cells in a particular sample, whereas non-PMA samples, equally 21 samples, which are not treated with PMA, means they contain both viable as well as dead DNA. Now what we did, we mapped these reads onto the genome of Enterobacter bugandensis to estimate their abundances. And what we got across the 3 flights or 3 time points, Enterobacter bugandensis was there. And in the flight 3, it was the most amount, but however, it is there. The concerning fact here is you can see the mean across all these time points are same in case of both non-PMA as well as PMA. That indicate the amount of reads of Enterobacter bugandensis that we are seeing are all viable. They are not dead. So this is an interesting fact that we observe. And then the most of the reads we noticed in the location 2. However, it is present across all the locations that we noticed. Then we try to say, okay, this is very much present there. So what all the microbes this is interacting with them? How is the microbiome landscape, whether Enterobacter bugandensis is manipulating the microbial community or not? So we constructed this network on what all the microbes present, where these big nodes are here, the location of the International Space Station, and what all the microbes that is present there interacting with the Enterobacter bugandensis. And we can see a group of Methylobacterium which is present in arid, as well as Pantoa is a group of Pantoa species there. We have some of the Klebsiella and other Staphylococcus and other Enterobacteriaceae organisms there. So given these organisms, we try to see the metabolic interactions of these organisms with the Enterobacter bugandensis in particular. So for that, we got the genomes of these organisms, which is, we put a cutoff of 1% relative abundance, and we got 43 organisms of that kind, and we got their genomes. We annotated these genomes, we constructed genome scale metabolic models, and then we did some simulations and got a metabolic network that represent the metabolism of these organisms. And then we simulated these organisms to see their metabolite exchange under minimal medium, considering that this environment of International Space Station will be not having a rich nutrient, and it will be mostly minimal kind of environment where they have sort of cleaning procedures there. So we simulated them under the minimal medium. And what we calculated here is, let's say this is a metabolic network of a particular organism, where in presence of certain metabolites, you can see certain pathways are active, whereas this side of pathway is not active, or we call it as a stuck reactions. So this is a bipartite network where these are the metabolites, where these square boxes, square nodes are the your reactions. Now in presence of another organism B, which produce some metabolite M2, which can be taken up by this organism, and certain stuck reactions are getting relieved here. So this organism B is helping A to do some more metabolic procedures. And we quantify this as a MSI, or we call it metabolic support index, which is essentially the number of stuck reactions are getting relieved when they are in a paired than its wild type condition. So we calculated that for all the organisms Enterobacter interacting with, and in the left-hand side blue panel, you can see this is the metabolic support Enterobacter giving to the other organism, and the more is the deeper darker color is the more number of metabolic support index, and very surprisingly we can see that these gram-positive organisms are getting more benefit here. Whereas on the right hand side, you can see whatever is Enterobacter bugandensis is getting from other organisms. So that is not much here. So indicating that Enterobacter bugandensis is probably a contributor to the community, and it's not getting much benefit. So this is a network-based approach, as I showed, and then we've moved forward to a constraint-based approach, and we do steadicom to see the growth of these organisms, and if there is a growth difference, we address this arrow or different ecological interactions. So in this study, when we see an arrow inward, that means Enterobacter bugandensis is getting more benefit, or essentially playing as a parasitic or amencellalistic organism. And with all the pantoas, that is another group of organisms that I showed is interacting with Enterobacter bugandensis, you can see the most of the arrows are towards inward, saying that Enterobacter bugandensis is a parasitic organism for this pantoa group of organisms. Whereas in the second, another group of organisms, which is Methylobacterium, where you can see the arrows are mostly outward. That means Methylobacterium is acting as a parasitic here, and is potential to kill the pathogen Enterobacter bugandensis in this particular environment. We also saw the interactions of gram-positive organisms that we mentioned getting a more benefit from the Enterobacter bugandensis, and that here you can see similarly that arrows are mostly outward. So that shows that Enterobacter bugandensis is regulating the microbial communities across the International Space Station. So given that, I would like to summarize what I described, that Enterobacter bugandensis is genetically, the strains we obtain from International Space Station are genetically and functionally different from what we see in the Earth. And then they are viable, and they mostly, we have seen them in the flight tree. The interactions particularly were mostly negative. We saw most of the parasitism, whereas where Enterobacter bugandensis predominantly acted as parasitic organism, whereas there are positive interactions but very less in number. And Methylobacterium being one of the organisms which is parasitic to Enterobacter bugandensis and have the potential to kill Enterobacter bugandensis under these circumstances. And particularly Methylobacterium phyllospiri had the most benefit out of this interaction. And what are the relevance of this study is Enterobacter bugandensis or any other pathogen, what is the genomic adaptation they had to survive in an extreme environment, and this could potentially be helping us to develop targeted antimicrobial treatment. And secondarily, there is the persistence and succession pattern of a pathogen in such certain space, closed space like International Space Station, which can be extended to other clean rooms like spacecraft assembly clean rooms, or some space like hospitals, where we see operation theater and ICU, which are closed space as well as having a lot of pathogens. And the most interesting part of this is the integration part, where we integrated genomics, metagenomics, as well as modeling to understand the microbial dynamics of this pathogen, and which can be extended to any other environment with any other organism to understand the microbial ecology of that particular landscape. So with that, I would like to thank the MVIF whole team as well as the audience. And this is our beautiful campus. If you are visiting India, please do visit us.